Hi. So in our last discussion, we saw that how a speciation happens, how a new species is formed, depends crucially on which definition of species we are talking about. And we specifically said that in the context of the speciation research, much of the emphasis is on the biological species concept, which means that speciation essentially becomes how reproductive isolation comes into being. So, in today's discussion, we are going to discuss the various mechanisms of reproductive isolation, and in the next discussion, we are going to look at in what ways those mechanisms evolve. So, this particular treatment is based after Fukuyama and Kirkpatrick's 2022 edition of the book Evolution, the textbook. So, before we dive into this, what exactly do I mean by reproductive isolation? So, reproductive isolation is the phenomena by which members of different biological species, remember that concept, they are prevented from producing offspring, or it ensures that any offspring that are produced are sterile. Now, the mechanisms, individual mechanisms that lead to this, these are either known as isolating mechanisms or they are known as reproductive isolating barriers. And today we are going to look at the various kinds of reproductive isolation mechanisms, and these can be grouped in many ways, but I will be following a particular grouping which is given in the Fukuyama and Kirkpatrick book, which itself is after Cohen and Orr's book called Speciation. It is a very nice book, very comprehensive treatment of the entire process of speciation, slightly old, about 21 years old, but even today the examples that they give and the way they weave the entire thing is just masterly. So, according to the classification that Cohen and Orr came up with, you can divide these reproductive isolation mechanisms into three major groups. So, the first, there are two major processes, or rather two major groupings, prezygotic and postzygotic, and prezygotic itself is divided into two parts, pre-mating and post-mating. So, that makes it three groups. So, we will start with the first one, which is pre-mating prezygotic barriers. Essentially, these are features that will impede or that will cause trouble with the transfer of gametes to members of other species, one way or the other. So, the first way in which this can happen is through time, temporal isolation. So, suppose you have multiple species which otherwise can exchange genes, but simply because they are not together at the same time, they are not able to exchange genes. So, I will give you one example. So, this is a bird, a small seabird known as the band-rumped storm petrel, and you find it in many places, but the one that we are talking about is in the southern hemisphere. So, in as you can see that they are found throughout Atlantic and Pacific oceans. So, in the southern hemisphere there are at least 5 archipelagos where these individuals, all of them are there at the same place, but there are 5 populations which are at different times. Okay. So, some population is at one time of the year, the other population of the same species is at the other time of the year, and DNA sequencing study by these people, Friesen et al., they showed that at least on 2 of these places, because these populations of the same species are coming at different times, there has been no genetic exchange between these populations for a very long time, okay, for some millions of years or something like that. I am not 100% sure about the timing, but for a very long time there has not been any genetic exchange. So, this is one example of how species or organisms can be at the same place, yet they never meet each other, and therefore, they are never able to exchange genes. The second thing that we can talk about is habitat isolation. Okay. They are geographically in the same place, but because of whatever reason their habitats have become isolated and therefore, they are not able to meet. So, here is an excellent example of that. So, these are two different species of ladybird beetles, you find them in Japan. So, the first one, Epilacna nipponica, that grows on thistles, this one, and the second one, Epilacna yasutomai, that grows on this plant called blue cohosh. Now, although in this picture the left one looks larger than the right one, in reality it is the other way around. It's this one is much larger in size than this one. So, although visually they are looking similar, if you find them side by side, you can clearly see the size difference. Now, turns out that both the species, they feed and mate on their respective host plants, and they are isolated only by this factor and nothing else. So, these guys, they will not go to that plant and vice versa, and that is it. If you actually take them to the lab and you actually put them on the same host plant and you make them mate, they can very easily mate and they can very easily produce fertile offsprings. But because the fact that in nature these two plants are separated away from each other and there is not much dispersal happening, that is how these plants, these beetles remain separated and they remain two species. So, this is the second one. The third one is what is known as immigrant inviability. So, in this what happens is that you have populations of the same species which get specialized to their habitats, and therefore, when they go to a different habitat, although they are capable of mating with those organisms, with the populations there, but when they go to a new habitat, they are not even able to establish themselves in that habitat, and therefore, they do not get a chance to mate. So, I will give you one example. So, there is this gall wasp called whatever Bellonocnema trite, and this thing, it grows on oak trees and it forms these, can you see these green ball like things, these are known as galls. Okay. So, it forms these tumor like galls on oak. Now, turns out that this wasp can grow on 3 different species of oaks, okay, 3 completely different species, and they end up specializing on their hosts. Okay. Now, if you take the females from one oak species of the same species of gall wasp, if you take them from one oak species and put them on a different oak species, then the females and the offspring, they are unable to initiate this gall formation. So, they are able to go there, but they are not able to initiate the gall formation, and because they are not able to initiate the gall formation, therefore, they are not available for mating with the local males, and as a result the populations of the same species which have ended up becoming specialized on the 3 different oak species, they cannot exchange genes with each other. They are simply not able to find each other to mate. So, that is one kind of thing. The other kind of isolation mechanism that happens is what is known as sexual isolation. And sexual isolation simply means that the males and the females, they are not able to mate with each other properly or not at all, depending on various kinds of cues. So, the first kind of cue, which is a very common kind of cue, is sound. You know that many organisms, they have special mating calls, and if the proper mating call is not heard, that is the conspecific mating call is not heard, then the female will simply not respond. And a big example of this is this Tungara frog. So, as you know that frogs, the males, they give these mating calls. This one is special because of this pouch that you can see. It is very funny. There are lots of videos of this on YouTube. You should check them out. So, turns out that the female Tungara frogs, they are going to respond only to exclusively to their conspecific mating calls. Any other mating call is constantly ignored. Another major cue for sexual isolation can be chemicals, pheromones, sex pheromones and so on. So, again many organisms tend to use it, particularly moths, it is very well known. So, for example, here is a particular species of moth. These are known as the European corn borers, and there are 2 different forms of the same moth. They are the same species, but turns out that they end up producing slightly different chemicals, and because of that So in this particular case it is the female which produces the sex pheromones and the males respond to them, and because these two forms of the same species produce slightly different chemicals, therefore the males do not respond to, or rather the males do not respond to the other females pheromones, and therefore these two end up becoming sexually isolated. The third form of sexual isolation, the third cue, is visual. This is extremely common. So, as you know that many organisms, for mating purposes there will be courtship that will be done by the males. We talked about it when we were talking about sexual selection. And if the courtship is not proper, or if the one of the sex is not displaying the appropriate visual cue, the other sex will simply not respond. So, just to give you one such example. So, this is a butterfly known as Heliconius pachinus, and these are found in Costa Rica and Panama. Now, as you can see that this has some very visible, very remarkable patterns, and the males recognize the females, it has been shown, by their wing color patterns. So, if you just change the wing color patterns, and most heterospecifics will have different wing color patterns, the males will simply refuse to recognize them. So, this is about sexual isolation. Another kind of stuff that primarily happens in plants, and that is pollinator isolation. So, you know that many plants, they depend on other animals for pollination, and many of these pollinators are actually extremely specific. In other words, they will pick up the pollens from one species and they will deposit it only in the same species. Which basically means they will only visit one species of flowers and not another species of flowers, and because of that the pollen never gets transferred across species. So, I will get you one example. So, there is this stuff called monkey flowers, two species, Mimulus cardinalis and Mimulus lewisii, and one of them, cardinalis, you can see that it is pollinated by hummingbirds, whereas the other one, lewisii, this is pollinated by bees. And turns out that if you physically take the pollen from one species and put it on the other, they can form, they can fertilize, fertile offspring can be formed. So, potentially they can interbreed with each other, but yet because their pollinators are extremely faithful. So, hummingbirds will not visit lewisii and bees will not visit the other one, cardinalis. Therefore, they end up not being able to exchange pollens, and that is why there is no gene flow between them, even though they are interfertile. So, just to summarize these 4-5 things that we talked about. So, we have the pre-mating pre-zygotic barriers. You can also subcategorize them in terms of ecological isolation, where the potential mates do not meet each other. So, this can be in terms of time, temporal isolation, or in terms of habitat, or in terms of immigrant inviability. And then you can also have a situation where the potential mates meet, in the sense that they are in the similar geographical area, but they are not able to mate, or they do not mate, whatever, and this can happen either because of sexual isolation due to various kinds of cues, we talked about 3 of them, or in the case of plants it can happen because pollinators are extremely species specific or plant specific. So, this was about pre-mating pre-zygotic barriers. Now, in many cases the mating does occur, but the zygotes are not formed. So, these are the post-mating but pre-zygotic barriers, and we will discuss a few. So, one of the things that we know is that there is a huge diversity in terms of the structure of the genitals of different species. So, for example, I am just showing you a very well known case in insects, that of different species of Drosophila. So, this figure over here, this shows you that inside, this is the male genital organ, and inside the male genital organ there is this thing known as the genital arch, and this genital arch is very important because it is involved in transferring sperms to the females. Now, what I am showing you over here is the structure of the genital arch in 3 different species of Drosophila. So, this is Seychellia, this is Mauritiana, and this is Drosophila simulans. And as you can see that there is quite a bit of difference between these 3 structures, right. In fact, this is the way in which these 3 species can be identified from each other. But if the genital structures are different, then how does it work? What exactly happens? So, it turns out that there are 2 things that can potentially happen over here. The first thing, of course, is that if the genital structures are different, then that can prevent the insemination or copulation simply because of mechanical incompatibility. Okay. So, you can think of one genital as the lock, the other genital as the key. So, until and unless the lock and the key match with each other, there won't be transfer of gametes, and because the 2 species mostly or many a times will end up having different kinds of genital structures, even if they come close to each other and even if they try to mate, they will not be able to physically initiate the copulation. The other situation that can potentially happen is that even if the lock and key are matching, even if they are able to initiate copulation, something does not feel right, so to speak. In other words, as long as the genitals are not appropriately complementary to each other as in the same species, the female or the male is going to have a physiological or a behavioral response, because of which there will typically be premature termination of copulation. So, even if copulation initiates before the sperms are transferred, or the copulation will break up, or in some cases because of mismatches, even if copulation happens, there are post copulatory reproductive fitness issues. So, these 2 mechanisms are not exactly antagonistic to each other, they are not mutually exclusive, and sometimes they can work in tandem. In other words, so sometimes what can happen is that even though the lock and key are not matching properly, the organisms can continue copulation, but even after copulation the amount of sperm transfer is not 100%, or even if the sperm transfer is happening, something might end up blocking after that. So, just to we have already given examples of that, but even in the case when the transfer of sperms have happened, there are many situations where the gametes are not able to fertilize each other, and this is what is known as gametic isolation. So, I will give you one very well known, well studied example. So, these are what are known as the abalone snails. They belong to a genus called Heliotis. About 70 species are known in this particular genus, and they are found pretty much all over the world. Now, these are external fertilizers. So, what that means is that the male releases the sperm into the water, the female releases the eggs, then the sperms and the eggs, they need to find each other, and the sperm needs to fertilize the egg. Now, there are 70 species, external fertilizations. Obviously, multiple species can and do exist in the same area. So, potentially the sperm of one species can end up finding the egg of a different species. However, these guys have a very peculiar way by which they are able to prevent fertilization of heterospecific gametes. How does that happen? So, it turns out that fertilization in this organism, it requires the interaction between 2 proteins. Why is that so? That is because the egg cell, since this is external fertilization, these are not sitting there unprotected. So, on top of the egg cell there is a proper proteinaceous layer, and the sperm cell needs to bore through this proteinaceous layer in order to access the egg and fertilize the egg. So, for this to happen, the sperm produces a protein known as the lysine, sperm protein lysine, and the egg has a protein which is known as VERL, vitelline envelope receptor for lysine. Now, these 2 proteins, they need to interact with each other, only then the sperm's lysine is going to be able to bore through that layer. Now turns out that these guys, the Haliotis snails, they have somehow evolved extremely specific interaction between the lysines and the whorls at the species level. In other words, the lysine of one species can interact only with the whorl of its same species, it cannot interact with the whorl of another species, and because of that it is not able to bore into the egg and cause fertilization. So, this is one way by which the gametes are able to, even if they come close to each other, they are not able to fertilize. This is a case wherein up to the protein level it is well known how gametic isolation happens, which is why I am showing you this case. But in general, gametic isolation is pretty well known across many taxa. So, just to summarize the post-mating pre-zygotic barriers, you can have mechanical isolation, reproductive structures of the sexes simply do not fit, or you can have copulatory isolation, even if they somehow are fitting with each other, behavioral or physiological responses of the sexes are preventing a proper copulation or transfer of sperms. And in the third one is gametic isolation, where even if the sperms or the ova are coming close to each other, they are not able to fertilize each other. So, we are done with the pre-zygotic barriers. Now, we are going to look at the post-zygotic barriers. What happens after fertilization has happened? So, again in this particular case what happens is that the hybrids are formed, but for whatever reason they have reduced fitness, and this reduced fitness can happen in many ways. So, we will just talk about 2 or 3. So, typically this is subdivided into 2 classes. When the hybrids fitness reduction is due to the external environment, it is called as extrinsic, and when the hybrid's fitness reduction is due to an internal property of the hybrid, that is when it is known as intrinsic post-zygotic barrier. So, let us look at them one by one. So, the first form in which there can be extrinsic post-zygotic incompatibility or isolation is what is known as ecological inviability. In other words, the hybrids are formed, the hybrids by themselves are fully functional, but because of some properties of the hybrid, they are going to have reduced fitness. So, I will give you one example. So, there are 2 species of heliconius butterflies, which, as we know, they are toxic. In other words, if the predator eats them, it either dies or most of the cases it has to end up vomiting the whole thing. Okay, that is, it creates a physiological reaction in the predator, and because of this they are distasteful. These butterflies are distasteful to the predator, and hence the predators end up avoiding these butterflies very religiously, and it has been shown that the predators, they identify these butterflies through their wing patterns. Now, turns out that in nature these 2 butterflies actually hybridize rather readily. So, they will, the males and females of the 2 species will happily mate with each other and produce babies, but the intermediate forms actually look very different from either of its parents, right. So, what Merrill et al. Did was they took models, small butterfly shaped models, which had the color patterns of either 2 parents or the hybrid, and placed those in forests. And what they wanted to check was what is the rate at which predation is happening on these. And when they did that, so here is one parent, here is another parent, and here is the hybrid. And as you can see, the 2 parents look very different from each other, and the hybrid is somewhat intermediate between the 2 parents. Now, what will happen? Remember, the predator's avoidance of these butterflies, that is a learned behavior, and therefore, for learning to happen, the population size has to be really high. If the population size is not high, then the predator will either not learn or it will learn, but it will forget very fast. Now, both these parent species, they are there in large numbers. Therefore, the predator's avoidance behavior is learned quickly and is maintained faithfully for a long time. But whenever you have hybrids forming, then typically the hybrids are going to be in a much lower number, and these guys, they are rarer in the population, and that is why the birds, first of all, they do not recognize them as distasteful, they end up attacking them, and because the learning is not reinforced, therefore, they keep on attacking them. As a result, you can see that the overall attack rate on the hybrids is almost double of the either parents, as a result of which the fitness of these hybrids is much lower. They basically they get eaten out. Okay. So, this is an example wherein the although physiologically the hybrids are perfectly well functioning, they are simply in a wrong environment, so to speak, and therefore, they end up getting selected against. The other way in which the ecology of the organism can reduce fitness is through what is known as behavioral sterility. So, we know that for mating to happen, there are lots of behaviors that are very important. If the male or the female are not behaving properly, copulation will not happen. Now, in many cases what happens is that the hybrid, although it is physiologically fine, it does not have the appropriate behavior, and because of that it fails to secure mating. So, I will give you one example. So, this is one species of grasshopper called the bow-winged grasshopper, and this is another species called the common field grasshopper. Now, we know that grasshoppers, they have these mating calls, and we also know that the mating calls of each species are very specific. Now, that turns out that the mating calls or the songs of the hybrids are actually intermediate between both parental species, and the male hybrids, they are actually behaviorally sterile, although physiologically they are perfectly fine, they are behaviorally sterile because all females, okay, either the parents or the offspring, I mean the two kinds of parents or the hybrid females themselves, none of them can recognize that particular song, and because of which these males, the hybrid males, they fail to get any kind of mating and they end up becoming behaviorally sterile. So, the other way, as I said, in which the fitness of the hybrids can be low will be intrinsic. So, it is not dependent on the external features, and the most interesting thing or the most common thing that often happens is what is known as hybrid inviability. In other words, the zygote is formed, but the zygote does not it is not born, it is either not born or it dies very young. So, many examples. I will just give you one example from the Drosophila literature. So, this is one species of Drosophila melanogaster, another species Drosophila simulans, sister species, closely related species. Now, turns out that if you take Drosophila melanogaster females and cross them with Drosophila simulans males, then the hybrid males will die as larvae. But if you do it the other way around, you take Drosophila melanogaster males and cross them with Drosophila simulans females, then it will be the hybrid females which will die, and they will die as eggs itself. They will not even hatch to become larvae. So, this observation comes from the work of Sturtevant. I am sure all of you remember Sturtevant. This is the person who came up with the concept of linkage as a 18-year-old undergraduate in Thomas and Morgan's lab. So, what Sturtevant showed is that intrinsic hybrid inviability can be complete, or sometimes it can also be partial. Okay. That basically means that not all the hybrids die, but a large fraction of the hybrids die, because of which the fitness is very low. And it can also be sex specific, which means sometimes the male sex gets affected, sometimes the female sex gets affected, sometimes both of them get affected, and it can also occur at different life stages, as I am showing you over here. Sometimes it is eggs, sometimes it is a larvae which die, and not coming from this study, but in general it also can be asymmetric, which basically means that, okay, also coming here actually, that if you cross the males with the females of in one combination, you will get one result. You take the same two species, but do the other way around, females of one and males of another, you end up getting a slightly different result. So, this, I mean, you can also, if you remember when we were talking about ligers and tigons, okay, not in the context of inviability, but we saw that you take male of which species and female of which species, that ends up giving you slightly different results in terms of what is the nature of the hybrid that you are getting. And what this is showing is that that can also have an effect in terms of the rate of the viability. In some cases you can get complete viability in one type of cross, but inviability in the other type of cross, or vice versa. Okay, so this is about hybrid inviability. The other thing that can happen is that the hybrid is viable, it grows, but then it turns out to be sterile. And of course, the most well known example of that is the mule, which is, as we know, obtained by crossing a horse with a donkey. Now, sterility again, like viability, can be total or partial, which means that either they can fail to produce any offspring, or the hybrids can have the ability to produce offspring, but that ability is seriously reduced. That is partial sterility. The sterility can be due to multiple reasons, and these can include things like if the two species, they have, let us say, chromosomal rearrangements. So, for example, if you remember, we discussed the fact that sometimes the one chromosomal segment can simply get inverted, that is what is known as an inversion. Or sometimes what can happen is that in one species, let us say some chromosomes, one segment from one chromosome and another segment from another chromosome, that have been interchanged, that is reciprocal translocation. Now, suppose you have two species, otherwise they are very similar, sister species, let us say, but in one case there have been inversions and in the other population there are no inversions. So, now when these hybrids are formed, and then when the hybrids are trying to reproduce and meiosis is trying to happen, then at the places, I mean, one set of chromosomes will have the inversion, the other set of chromosomes will not have the inversion or translocation, whatever. In those cases, the binding of the homologous chromosomes for the sake of meiosis can become seriously affected. And if that effect is severe and meiosis ends up failing, then that can be one way in which sterility can be induced. The same thing can happen if, let us say, the two species have slightly different genes, and these genes produce proteins which end up having an interaction with each other, because of which the cell is not able to survive. Or one of the most common ways in which this happens is if the two species have very different chromosome numbers, then their hybrids, when they are formed, there will be some chromosomes which will not have a homologous chromosome to pair with, and because of that, when meiosis happens, you are going to have gametes with very different chromosome numbers, which again might end up leading to a complete breakdown of fertility. So, this is about the postzygotic barriers. Just to summarize it for you. So, it can be either extrinsic or intrinsic. Extrinsic, when the low fitness of hybrids is due to environmental reasons, that can be either due to ecological inviability or behavioral sterility. And when it comes to intrinsic is when the low hybrid fitness is independent of the environmental context, and it can be either due to hybrid inviability or hybrid sterility. Now, before we finish this discussion, I would like to make 3 observations about reproductive isolation. So, the first thing is that although we talked about them as a list, it is not at all essential that they are mutually exclusive. These various mechanisms are mutually exclusive to each other. As a matter of fact, we know that in many cases, for any two species, multiple of these mechanisms might be operating simultaneously. So, just to give you one example, we will go back to the monkey flowers that we talked about. So, if you remember that these are two different flowers and one of them is pollinated by hummingbirds, the other is pollinated by bees. Now, turns out that in this particular species, generally this one, this lewisi, that occurs at a higher elevation than cardinalis, and there are some places where they do co-occur, but if you forget about the co-occur part, much of the isolation between these two species is simply because of the elevation alone. In other words, because there are the populations which are at two different elevations, for those the fact that they are not together is sufficient for no gene transfer to happen. Now, if you look at only that subset which is found together at intermediate altitudes, there 98% of the separation is because of the pollinator preference which we talked about. Okay. So, cardinalis is only and exclusively pollinated by hummingbirds which do not visit the lewisi, and the bees that visit the lewisi, they do not visit the cardinalis. So, that is the primary mechanism for when they are co-occurring. But even on top of that, if you actually end up trying to mate them, there are three other things which can still end up reducing the probability of gene flow. So, for example, if you put the pollens of both species simultaneously on the pistils of these flowers, then the conspecific pollen has a much greater chance of fertilizing the ovule. So, that is what is known as pollen precedence. Similarly, even if let us say fertilization has happened, the germination success of the hybrids is slightly lower, and even if germination has happened, the fertility of the hybrids is slightly lower. However, if you actually end up seeing how important these things are, these last three things, they play a small role even if they exist along with the two big ones. So, the main point I am trying to make here is that multiple mechanisms, reproductive isolation mechanisms, often end up working in tandem with each other, which makes the gene flow between these things even more difficult in practice. The second thing that I want to talk about is a observation by JBS Halden. We have talked about Halden, if you remember, which is known as Halden's rule. What is Halden's rule? So, Halden said that if two species or two populations mate and there is hybrid sterility or hybrid inviability, then typically that is limited to the heterogametic sex. Now, what do I mean by that? So, if you remember, in human beings and in all mammals, we have the XY system of sex determination. So, the females are all XX and the males are XY. So, that is why the males are the heterogametic sex, they produce two different kinds of gametes, whereas the females are the homogametic sex, they produce only gametes which contain the X chromosome. Now, you have a similar thing in birds where it is actually reversed, where the females are heterogametic and the males are the homogametic sex. So, Halden's rule suggests that hybrid sterility or inviability will be limited to the heterogametic sex, which means that primarily males in mammals, but primarily females in birds and butterflies. Now, turns out that this is actually rather widely observed in many systems, although there are one or two exceptions known here and there. So, for example, one of the exceptions is something that we just talked about a few minutes back, which is in the context of Drosophila. So, if you remember, I showed you that Drosophila melanogaster males, when they mate with Drosophila simulans females, then the hybrid females die, and yet this is a case where Drosophila is heterogametic like us. So, males are XY whereas the females are XX. So, it should have happened in the males, but in this particular combination it happens in the females. But such one or two exceptions notwithstanding, Halliday's rule in general is widely observed. And the final thing that I want to talk about is the speed with which reproductive isolation ends up setting in. Now, turns out that there is a huge variation in the time taken for reproductive isolation from setting in to it becoming a rather strong factor. Now, as we saw, reproductive isolation in many cases is not 100%. That is why I am not saying it going from 0 to 1, but I am saying starting from when it started to evolve to the point where it has become really strong mechanism. So, for example, we have already seen that when speciation happens due to polyploidy, as it happens in many plant species, then that obviously can happen within a generation very fast. In many cases, it has been shown that you can start with a single or a few ancestral forms, and then over a relatively short span of time, these can rapidly diverge into many species, and this is what is known as adaptive radiation. So, just to give you a very famous example of adaptive radiation, there is this very famous and huge lake in Africa called Lake Victoria, and in that lake it has been shown that about 15,000 years ago there were 5 ancestral species of a particular kind of fish known as the cichlid fish, and over just 15,000 years we have more than 450 species which have evolved, which means that an average time of about 2,300 years per speciation, okay, about 2 millennia, 2.3 millennia. That is actually extremely rapid, and that sits on one side of the scale, adaptive radiation examples. On the other side of the scale, we have things like sister species of Drosophila. So, for example, Melanogaster, Simulens and Mauritiana and all those things that I was talking about. Contemporary genetic estimates suggest that they have taken about 200,000 years to evolve from one species to another. If you look at birds, okay, for example, prior to birds, on an average across all taxa, typically the estimate is that if you have populations which are geographically isolated, which means that geographically they are not even exchanging genes, then in order for them to evolve total reproductive isolation, it takes between 1.1 to 2.7 million years, that is 11 to 27 lakh years, and that obviously is a huge number. But if you look at birds, it has been shown that some bird populations that have evolved or diverged from each other more than 5 million years ago, even today if you bring them together, they are able to exchange genes and are fully fertile. And if you think birds are an exception, look at plants. It has been shown that there are some trees which can still form hybrids even though they diverged from those species between 10 to 50 millions of years back. So, all this is telling us is that the process of reproductive isolation is not a very simple process. There has to be some variation in them, and the way they set in, okay, is not how do I say, there is a lot of complexity in the way they set in. And in our next discussion, we are going to exactly look at this particular question: what is the way in which these reproductive mechanisms of reproductive isolation, what is the way in which they end up evolving. So, see you in the next discussion. Bye.